I'm Jonathan Javich from Area Neuroscience and Molecular Therapeutics. I have the honor of introducing Dave today. But first, I'm just going to let you know about the Grand Rounds presentation for next week, Wednesday, January 13th, which is Health, Equity, and Justice for People with Mental Illness by David Henderson, who is the professor and chair of psychiatry at Boston University. Today, we have our own David Sulzer presenting. Dave's a professor of neurobiology in the departments of psychiatry, neurology, and pharmacology at Columbia, and a research scientist at NISP. He attended Michigan State University, where he started his work on plant breeding and genetics, later received his PhD in biology from Columbia, where he studied with Dr. Eric Holtzman. Did his postdoc with Stephen Rayport at NISP, where together they made important contributions about our understanding of how amphetamine works in the brain, and also to co-release of dopamine and glutamate from dopamine neurons. His lab has published over 200 studies on synaptic function in normal and disease states, been cited over 40,000 times. And the depth and breadth of his work is recognized by his many longstanding grants from multiple neuroscience-related institutes, NIMH, NIDA, and NIMDS, as well as a number of private foundations. He's the founder of the Dopamine Society, the Gordon Conference on Parkinson's Disease, and Nature Parkinson's Disease Journal. He's received awards from the McKnight, Simons, Helmsley, Narsat, Huntington's, and Aaron Diamond Foundation. Dave's trained a whole generation of graduate students and postdocs who've gone on to have successful careers in their own right. And it's a pleasure to have him here today to update us on some of his work. His title is The Dopamine Synapse Learning and Psychiatric Neurodevelopmental Disorders.
Okay, great. Thank you, Jonathan, Jeff, Simon, and John. You all hear me fine? Okay, yeah. So it's nice to be invited to give the first Grand Rounds of the year, and particularly on what looks to be an auspicious historical day for our country. Just a little bit of lab introduction beyond what Jonathan just said. We moved to NISP 4 years ago now this month. Our research is on synapses, particularly in the basal ganglia, in normal learning and in psychiatric neurological disease. We use approaches from a variety of disciplines. The lab's been busy, as Jonathan said, with over 200 peer-reviewed papers in this work. But besides our work in the ivory tower on neuroscience stuff, we're actually taking some of those findings and moving them into the clinic. Here's a list of several of the ongoing clinical projects we have with different members of NISP. For those of you who are interested in the work we'll be talking about today, during the pandemic, we have virtual lab meetings Fridays at 10:30. If you're interested, send me an email, and we welcome people to hear about all this work in more detail. I do not have any conflicts of interest to report. So what we'll be talking about today is sort of a neuroscience basis of something that's interesting to everybody in the psychiatry department, how you become who you are, your personality, your learning. Psychologists sometimes call this actor evaluator pathway. This young violinist is trying different things out with her motor system, and sometimes the audience likes it, other times they don't, or it's her own evaluation. And eventually you go through a process that our lab calls reiterative synaptic selection, and one day you end up being Jascha Heifetz or somebody like that. So I'm going to divide this into two sections. The first is the longer one. And you get neuroscientists coming in here a few times a year to give grand rounds, and they talk about worms and snails and fleas and flies, and and saying, oh, these synaptic changes alter behavior. And I don't blame you if you wonder, yeah, what does this have to do to what we really do? Does this actually work in higher animals? We're going to show you 3 stories from graduate students who say, yeah, this actually is happening. And in the second part, we're going to talk about how we can study the maturation of these synaptic changes and how these have interesting implications for the work that's done in the department, particularly in this case in neurodevelopmental disorders. So starting historically, the discovery of the reward pathway is one of the most interesting, uh, important and famous experiments in science from Jim Olds, who at the time was at McGill University about 60 years ago. And what he found was that if he allowed a rat to press a lever to administer current into the rat's own brain, that it would do this, it would start, maybe doing this 10 times an hour, and very quickly, within minutes, learn to do this self-administration 5,000 times an hour. Now, at the time this was happening, dopamine as a neurotransmitter was just being discovered by Kathleen Montague and Arvid Carlson. Dr. Olds died just at the time that he started to realize that dopamine, but he already knew from the beginning that was blocked by chlorpromazine, the antipsychotic that was already used at that time. One thing that people often don't mention from this original work is that a lot of the brain actually causes the animal to approach and hit this lever. And there's only a relatively small fraction of the area of the brain that causes avoidance of the lever. Now, we're going to show you the same thing, but it's just done with video now. So just does this video. This is exactly what Jim Olds was doing. It's a little disturbing to watch. So this mouse is administering electricity into his own ventral tegmental area and stimulating his own dopamine system. And he'll keep doing this until Avery disconnects the current from the electrode. So now very recently, we've started to understand the kinetics of the system much better. So I want to go into that a little bit with you. This is due to work by an outstanding postdoc in the lab named Maha Somayaji. So you may see her walking around in the halls. And what she's done is improve an old approach. So what she's doing is taking an anesthetized mouse and stimulating the VTA in the substantia nigra, where the dopamine neurons live, and then measuring the dopamine release and reuptake live with a electrochemical electrode. And we call this cyclic voltammetry. And the way this works is when you stimulate over here, you get a signal, and this signal is telling you the concentration of dopamine. In fact, this is telling you the concentration of dopamine, and this is telling you the rate of the reuptake by the dopamine uptake transporter, which is the site of action where cocaine, Ritalin, amphetamine, it all work. This new approach by Maha was just published this month in PNAS, so do check out the paper. Now, when she does this in a lightly anesthetized mouse, and what I mean by light anesthesia is they're getting some isoflurane, and if you remove the isoflurane, in a few seconds, they wake up as if nothing happened. And she can also record as well as stimulate these neurons. And what we see are these midbrain dopamine neurons are going through this regular so-called tonic or pacemaking activity. Now, this is interesting because it's unusual for brain neurons to be pacemakers. Your heart cells are pacemakers. You could take them out, put them in a petri dish, and they'll keep firing at the same rate. Dopamine neurons are one of those rare neurons that will do the same sort of thing. And so they'll have a pacemaking activity. It's faster than your heart. It's about 4 times a second. And now the language of the dopamine neuron is this pacemaking activity. And over that are bursts. And these, so I'm pointing out some bursts over here. And this bursting activity occurs at about 20 hertz. So you could say the language of the dopamine neuron doesn't have that much variety. It kind of goes between 4 hertz and a burst of 20 hertz, and it does this all the time. But the 20 hertz burst is actually how the neuron is communicating with the other cells in the brain. And we'll get into that, in just a little bit. So as Maha was determining how to do this approach, she's saying, all right, if the burst activity is important, how should we do this? And so she did a lot of, I think, well-designed, she designed it, not me, experiments to determine how we're going to do this. She looked at the frequency of the bursts and the number of pulses that would be in a burst. Remember that the animal, we now apply the burst rather than the animal doing it because it, say, found unexpected reward or something that surprised it in its cage. So now Maha is applying this, and so we choose these areas here where there's a big slope. Now, why do we do that? That's because if you want to look at something that's physiologically relevant, you want to look like a drug or a gene, a mutation in a gene. You want to look at where it's going to have the most effect. And that is almost always where physiologically relevant behavior is happening. So we do understand that. One slide of math, and you don't really have to look at it. But by using this mathematical approach, we really are starting to understand the basics of dopamine transmission. This is work by a computer science master's student, Shashank N, who's working with our lab. And Shashank has worked out a series of equations. This first one describes what happens to dopamine in the striatum. The second one is what happens to the electrode. The third one is where it gets interesting. This term allows you to derive things like facilitation and depression. So if you have a series of bursts, as you would in normal activity when you're going around your normal day, this will help say how one little epoch of activity is going to affect the next. And it works quite nicely. So the orange here is Maha's real data, and blue is Shashank's fit. This is a fit with a single burst. And from this, we can say how much dopamine is released per stimulus, in this case of substantia nigra and BTA. And we can get the kinetics of the dopamine uptake transporter, which is interesting because now we can actually get, usually do it by boiling some tissue and binding. We're actually getting it live in the animal. And we can get these numbers over here that tell us about this stuff. And perhaps more interesting is we can see plasticity now. So here's a series of bursts. Now, if this were real, the bursts would be here, but they'd be somewhat haphazard. Here we're controlling them. And look what happens. This is the dopamine release with the first burst. Here's the second, here's the third. This is a facilitation, okay? And we can figure out the facilitation. And the facilitation, there's a couple of depression. This facilitation happens with a time constant of about 6 seconds. And as you can see, you can fit it very well. So what's that due to? This ends up being very interesting, particularly for neurologists, because Maha showed that this facilitation is due to the protein alpha-synuclein. Now, you all probably have heard of alpha-synuclein. It is a cause of Parkinson's disease. The Lewy body is mostly made of non-degraded alpha-synuclein that builds up over the course of Parkinson's disease, and it shows up in all sorts of other dimensions as well. So you just going here, see, there's a facilitation in a normal animal. The facilitation is gone in a knockout animal. That means alpha-synuclein role. Now, interrupting myself, alpha-synuclein is 1% of brain protein. We know that it's an important protein, but we didn't know what the heck it does. My lab has been working on it as well as many other labs. We finally understand what it's doing. The only reason we understand what it's doing is because we're finally looking at physiologically relevant activity of the dopamine system, which we can't do with things like the brain slice. I'll just mention briefly, we do have, you can look at Maha's paper, you can also see my review with Rob Edwards, and that goes into how this actually works. Synuclein's effect is on the fusion and the unfusion, the recycling of the synaptic vesicle cycle, and moves the vesicles in and moves them out faster, and that's why you can get the facilitation. Now, again, this is not really simply ivory tower. For instance, most of the people who've been going to grand rounds for a while know that Anissa Abidagham's work and Mark Lorwell have for years shown that there was too much dopamine release, or abnormally high amounts of dopamine release in people with schizophrenia. We suspect that this protein and some allied proteins, some similar proteins, may be involved in this, and that's one reason we're working with Andy Dwork's lab at PI right now. Also, Maha's work is showing how Adderall and Ritalin, and for that matter, cocaine, and antipsychotics are actually working on the system in very different ways with different kinetics than people had thought before. So I hope that you'll consider inviting her for grand rounds next year, and she can go into more detail on this. Here's the first of the vignettes. I already mentioned Avery, he's a PhD candidate, and we're going to talk about a couple of kinds of learning important to everyone in this audience, Pavlovian and operant conditioning. What Avery's doing is very similar to what Maha's doing, but two big differences. One, this is a behaving mouse, not an anesthetized mouse. Number two, instead of measuring actual dopamine, what he's measuring are the activity of the dopamine axons. So remember, the dopamine neurons go through bursts. Now, when those bursts happen, there's a big increase in calcium in the axon, which we can measure using the fluorescent calcium reporter GCAM. So we put that in the dopamine neurons. And now he's going to measure the dopamine release during a Pavlovian task. And this mouse has learned that when he hears a tone, and when the tone plays, this light will go off so you can see when it comes on. Now, watch down here too as the mouse is behaving. You see these spikes of dopamine activity. Now, this animal has already learned that when he hears a cue, that can come over here and start sipping some sugar water. So what you see here is after the learning, that's when the dopamine axons get this big response. And then they get a second response as the animal is sipping the sugar water. So that's a very simple Pavlovian behavior. And it's showing that there is a change in these pathways with learning. So the first day, the animal has not learned that the sound cue means that sugar water's there. These are color charts. You read them, just read them across. Each one is a separate trial. So here's 20 trials. Red means a lot of activity of the dopamine axons, blue is low activity. After training, as soon as the animal hears, there's a big response. You just saw it to the cue. And following that, as the animal is sipping that sugar water, there's a continued response. So what I've shown you so far is really quite classical. It really goes back to Jim Olds and to people like Wolfram Schultz and Mark Whiteman and Regina Corelli have been studying the system for a long time. So we know already that dopamine is involved in things like unexpected reward and this kind of Pavlovian behavior. What's not been known is what happens next. So you release the dopamine, what the heck is it doing? So these circuits were described, I described them mostly to Mélanie DeLong, who's at Emory University. So here are the dopamine cell bodies. This, by the way, is from a new review by Stefano Cataldi in our group and on motor learning and the system. So you have these neurons releasing dopamine over here in the striatum. Now, they are converging with other excitatory inputs coming from the cortex, also from the thalamus and a few other regions. Then there are output neurons. So you do all these calculations in here. There are essentially two major output pathways. One's called the direct pathway, one's called the indirect pathway. Chris Kellendonk at PI, for instance, also studies these pathways, as do several other people. So there are outputs. This one is called the indirect pathway because it doesn't go right back to the dendritic area of the substantia nigra. This is called the direct pathway. The indirect pathway tends to have D1 type dopamine neurons and the indirect pathway D2 type. So we want to say, what happens during these behaviors when we know what's happening with dopamine? What the heck is that doing to control these next steps, the output nuclei of these pathway where all the calculation is done? So the calculation is done in these kinds of neurons, which are most of the neurons in the striatum. These are called spiny projection neurons, projection because they project out of the striatum, and spiny because here from Ori Lieberman's thesis, you can see these spines. So each of these spines is receiving input from the cortex, excitatory input. Now, they're funny neurons. They're not tonic at all. They're quiet. At least in the adult, they're quiet. You'll hear more about when they're young. So they're very quiet neurons, and they need a lot of excitation to fire. And this is going to be important for neurodevelopmental disorders. Now, let's look at what that synapse actually looks like, because unless you've studied this stuff, it's going to surprise you. This is an EM from Melissa Nuremberg, used to be in our lab. And here's a spine from one of the spiny neurons. Here's a cortical input to that spine. And over here is a dopamine varicosity full of dopamine synaptic vesicles. And this happens to be the dopamine uptake transporter. So let's just make this into a little cartoon and think about it for a second. If we just kind of draw this thing and we put in these three components, and we're going to add a fourth because the cholinergic interneuron is also very important here. Each of these has a receptor for itself and the other three neurotransmitter types. Now, if you're a neuromuscular junction, you kind of have an on and off switch, 0 and a 1. And you can change the frequency of your the compression of your muscle fibers, the contraction. But you can't really do much else in short term other than turn them on and off at different frequencies. This is a very different situation at what we're calling the striatal microcircuit. Just if you have the combinations here, instead of 2 to the 1, it's 2 to, we have over 4,000 different states. So I tell our grad students and postdocs, there are a lot of challenges to having a career in science. This is not one, because we're not going to figure, one reason we're so complicated is because this synapse is so damn complicated, and it's going to take a long time to work this out. A little more on the spiny projection neuron in the adult. So in the adult, they don't have burst firing, but they have another unusual quality. It's called bistability. And this was mostly worked out by Charlie Wilson, who's at the University of Texas, San Antonio. And what Charlie shows is that there's a down state, he calls this a down state, where the neuron is very hyperpolarized. And what happens down here is you will not fire an action potential from the down state. You just can't get activity. Now, if you get enough activity, you will eventually drive it to another stable, semi-stable resting potential, which is up here about minus 50 millivolts. Of course, he calls that an upstate. And from the upstate, you can start to fire bursts. So again, the language of these neurons is mostly in bursts. Now, why does this happen? This happens because of a particular channel you're going to hear more about. It's called a KIR, for K for potassium, IR for inward rectifying channel. Most channels are, most of these channels are opened by changes in voltage. Most potassium channels are turned on when you have a depolarization. In fact, that's what ends your action potential, because you open up potassium channels here when the cell is quite depolarized in an action potential. These are funny ones. In fact, it needs to be called the funny current. Anyway, so you have this downstate over here. The potassium channels are open here, and then they close as you get more depolarized. So this tends to clamp the cells and keep them quiet. So you can do a lot of stuff. You can walk around if you're in a mouse in your cage, and these guys are not going to fire unless you get a hell of a lot of excitation. That's in the adult. So now Avery is going to do that same Pavlovian behavior, but instead of measuring dopamine, he's going to measure the output neurons. So first, let's look at the direct pathway neuron. And here's the mouse walking around. Now remember, the mouse is going to get a cue over here in the time, and the light will go off and get a cue. Remember, the dopamine neurons became very active. What happens direct pathway? Nothing. They don't respond at all. They do respond when you go over and start sipping some of that sugar water. Okay, what about the indirect pathway? And I'll say briefly to the cognoscenti in the audience, cognoscenti, that this does not exactly go along with Mahlon DeLong's go and no-go pathways. It's becoming a little more complicated simply because we have techniques that Mahlon didn't have available to him at the time. So we're going to look at that same behavior. We've seen it with dopamine, we've seen the direct pathway, now we're seeing it with this indirect pathway that have the D2 receptors that antipsychotics work on. Now the cue is about to come on. Here comes the cue. What happens? Now we get a behavior. And then we get more behavior as the animal goes over and licks some sugar. So this is already a little bit apocryphal from the understanding. Now we can line these up and see something pretty interesting. After the animal has learned, which, several days of training, you saw that the dopamine will be turned on immediately after the cue. This precedes the activation of these two neurons, and it's probably required for this. It's something that Avery continues to work on. Let's make this a little more appropriate for psychiatry by looking at operant behavior, meaning that the animal has to say, okay, time for me to have some sugar water. In a way very similar to what Jim Olds did, except there's more timing. He has to go over there, hit a lever, and then walk over and take some sugar. So first looking at the direct pathway. Now the animal is going over there, hitting the lever. And when he hits the lever, we get an activation of the D1 pathway. Remember, we did not get this activation when the animal learned the cue. And then sucrose, and we get, as they're sipping sucrose, they get activation of that pathway. What about the indirect pathway, which was activated by learned cue? Well, no, it's very different. Here are the animals taking sugar. As it does, it activates this indirect pathway. But when it decides time to go over there, hit a lever so I can get some more sugar water, nothing. It's completely different than cued learning. So these guys all have different kinds of learning. And it's been short, but I've shown you a lot. I just want to repeat what I've shown you so far. This is all Avery's work so far. So there's an increase in dopamine axon activity when a learned cue, like sound, is paired with the reward, sugar water. Indirect and direct pathway neurons are both activated as the animal's sipping the sugar water, but the dopamine axon activity precedes a learned increase of the indirect pathway in response to the cue. In voluntary action, operant action, that doesn't happen at all. Then the direct pathway, and this is similar to Malon's idea of the go pathway, is activated when the animal does a voluntary action. So if you've been putting all this together, that leads to a real conundrum. Remember that SPN neurons, these spiny neurons, do not fire in the adult. They need inputs. And that input can come from the cortex or amygdala, hippocampus, the thalamus. But I'm telling you that the dopamine is controlling this. So how can that possibly be? So to do that, we're going to go to work that Nigel Bamford, who's now chair of pediatric neurology at Yale, but he was a postdoc in our group, and he did this work in several very good papers and continues to do this work at Yale. So the way that he designed a method to look at that is we just got our first two photon microscope, and he figured out how you can stimulate the motor cortex over here, where the cell bodies are, and then look at the motor stratum over here. So you can use one electrode to stimulate here, or you can use the second electrode to stimulate dopamine release over here, and you can look at the interaction. Or you don't have to use an electrode. You get exactly the same thing with cocaine, with amphetamine, and with the D2 agonists like Quinfol. And now we're going to see if this is going to work. This is why. It works. Simon, thank you. We have a wonderful person working on Zoom today named Simon Williamson, and this did not work before and it works now. So thank you, Simon. So what we're doing over here, this is one axon running from the cortex into the striatum. And so each of these are those cortical synapses in the striatum. And you could see that they're green, and that's because they've taken up an endocytic dye. And so the synaptic vesicles are now filled with a green marker. When the vesicle fuses, it's going to release this marker. And from that, we can get the activity of each of these synapses. Now, these are all synapses on just one axon. But as you'll see here, as I think you'll see here, they have different rates of destaining, which means they have different rates of synaptic activity. So even from this one axon, we can look at each of these and we can get the rate of destaining. So this is when it's full. This is when it's empty. That means the synaptic vesicles have fused. And we can look at each of these. One of them here never fused at all. It's a silent synapse. These other ones have different rates of fusion. And so this is a fast one. It's half of its content. This is half of the content at 200 seconds. This is a slow one that required 400 seconds for release. Now, where does the cortex work? I told you that dopamine neurons fire in bursts. That's where their activity is. It's about 20 hertz. Well, it turns out that for your motor cortex, 20 hertz is kind of the magic number 2. This is a nice paper. It's an ECOG paper that was done during epilepsy surgery. Very, similar to the Penfield surgery, but now done with the cortical electrodes. It's done for multi-electrodes. It's done for epilepsy. And the patient is awake, of course, during this, and you could say, move your right index finger. And as you do that, you look at the left index finger portion of the motor striatum, and you see that most of the neurons are firing at about 20 hertz. So voluntary motor action corresponds to about 20 hertz firing in the motor cortex. So now let's take all that work that Nigel was doing, looking at these thousands of cortical inputs into the striatum. And you all know that when you're asleep, you have a slow wave called a delta wave. It's about 1 hertz. the neurons are holding hands or swaying together, doing kumbaya. There's not a lot of personality in there. And now Nigel can stimulate dopamine, or he can not stimulate dopamine, and we can look at the distribution. So each of these is one synapse. The gray is without dopamine, the red is when he released dopamine, and you can see they mostly overlap. That means there's very little effect of dopamine. Now let's go to voluntary motor action at 20 hertz. Purple is without dopamine, this is with dopamine. Aha, enormous difference. Dopamine has an enormous effect on cortical inputs, but it has to be done, again, at physiologically appropriate rates. This is why this had not been seen before. Now, the old question is, dopamine excitatory or inhibitory? And we're going to say, no, that's not the right question. What it really is it's a filter. So it doesn't touch these very rapid, remember this fasty staining, it's very rapid synapses, but it slows down slower synapses even more. So it's what engineers would call a high pass filter. Now you could think about this and say, oh, we only get plasticity here if we have a busy cortex and we have a busy dopamine system, like unexpected reward. And you can imagine out of that how you can start getting a learning mechanism. For instance, that violin player a few minutes ago. Well, when things work, you just activated the heck out of the cortex, 20 hertz, and you just activated the heck out of the dopamine system, like we saw in those mice. And you start to bring things out. Here's the second story. I'll make it a little shorter than the first one. This is from Minerva Wong. Yeah, go ahead.
Before you get into that, going back to your
is this Jeff or who is this?
That's Jeffrey.
Okay.
Two slides previously. Are the cortical striatal inputs to the spiny neurons glutamatergic?
Yes, they're all glutamatergic, all of them.
And where are the cholinergic ones that you alluded to earlier coming from?
Boy, you should start joining our lab meetings, Fridays, 10:30s. They are interneurons, and they have both synapses and volume transmission.
Okay. All right,
and they're really interesting. And a couple of people in the lab, Avery and Ori Lieberman, are doing some really interesting work on them.
Yeah,
so,
I mean, I remember Jonathan in a paper we wrote for Trends in, I think, Pharmacology some time ago, alluded to that source of input as being relevant for dopaminergic transmission and antipsychotic therapeutics.
Well, antipsychotics is going to turn out to be interesting, but it is definitely appropriate. In fact, we discovered that in 2004. It's a paper by Wei Zhang. And yeah, the acetylcholine system is directly activating dopamine.
Well, you might keep that in your working memory, because there's a paper in press in New England Journal on that Karuna compound, the xanomeline M1M4 partial agonist. That's going to be coming out.
Very good. Yeah, well, even nicotine, smoking works on the system. And, invite me back or one of the lab members back next year, and they can go into a lot on this. But it's pretty interesting. I would certainly agree with what you're saying. Okay, if you don't mind, let's go on to a couple of more stories. This is a sensory motor behavior. Anders Borgqvist, who's now the professor of Karolinska, did this. Now, this is a corridor test, and the animal will take sugar pellets from either side, if they're a normal animal, left and right side, both. But what if you remove dopamine from one side? And that's over here. Now, you could do that with the toxin. And if you do that, the animal will only take sugar pellets from the side contralateral to the intact side of the brain, okay? Which is what this animal's doing. The only way you can get the sugar pellets from the other side of this cage is by turning around. And now we'll take pellets also from what was the left side, it's now the right side. So this is a way that we can study innervation of dopamine and loss of dopamine, even in the same brain. And this is what Minerva is going to do. And so first, just to show you the behavior, in an intact animal, this is left sugar pellets, these are right sugar pellets, and an animal will get them from both sides. You give them L-Dopa, which is the precursor to dopamine that they give to Parkinson's patients, does absolutely nothing. Now let's look at an animal that has lost its dopamine on the left-hand side. And so it is only accumulating sugar from the contralateral side to the intact side. which is over here, and they're not taking sugar pellets from the left at all. I'm sorry, from the right-hand side at all. You give them L-DOPA, and what happens is they actually overcompensate. Some of these animals down here are now only taking sugar pellets from the side that they had previously ignored. Now, just to cut to the chase for time, we can completely normalize this, and this introduces a new chemical into all this complicated circuitry, which is cannabinoids. So if we use a CB1 cannabinoid blocker, we can go back to completely normal behavior. Now, to do this, to study this, Minerva is using the same system that I introduced, that Nigel had invented. And you will remember that the fast synapses of the cortex and the stratum releasing glutamate, as Jeffrey just pointed out, are over here. And there's this straight line is a normal distribution. There's just normal distribution of cortical inputs. So what happens when we remove dopamine and then we come back and look at that side of the brain where the dopamine has been lost? All of those cortical inputs are slowed down. They're all slower. Now, that's not normal, because remember, we said what dopamine does is it doesn't just slow everything down, it acts as a filter. These fast guys down here, if this was a normal animal, would be faster. So what we can do is add L-DOPA. Okay, if we add L-DOPA, we bring them back to being faster again. So we have normalized them, but the filtering is still not here. The combination of L-DOPA and filtering, L-DOPA and the cannabinoid blocker, brings back normal filtering, even in a Parkinsonian or hemi-Parkinsonian animal, and again, brings them back to normal behavior. So what does that mean? What that means, and this is work from our lab, but also from David Lovinger's lab at NIH, and as I mentioned, Nigel has continued this in his own lab at Yale. If you release dopamine, say you have unexpected reward, and there's that mouse walking around, and you release dopamine, as we saw in that mouse, what's happening at the plasticity of the synapse, of the glutamate coming into the spine? And the answer is, it doesn't do a damn thing. There is no plasticity. What happens to plasticity if you just simply release glutamate in a relatively normal fashion, as you do all the time when you're doing, as I'm doing right now when I'm speaking, and you're doing this, you're listening, you're releasing glutamate onto these neurons, it's not having any plasticity. If you have burst firing, specifically burst firing by both of these simultaneously, which happens, remember, if you're doing something with the motor cortex and the dopamine system is being activated, what we believe is happening is you are stimulating the production of via two receptors. You need both of these activated, the metabotropic receptor and the D2 dopamine receptor. And you're making a cannabinoid. This happens to be an anandamide. And that feeds back onto this presynaptic CB1 receptor. And that's what's responsible for that inhibition of the dopamine, of the glutamate neurons. So this is a combinatorial effect. And again, you can imagine, this is really just a different way of the same story, how the combination of cortical activity and dopamine activity will now lead to plasticity. But they have to be coincident. Yeah.
The effect is an agonist effect on the CB1 receptor?
The antagonist effect is the one that brings it back to the, if you have antagonist plus dopamine replacement, that brings it back to normal. If you simply put on an agonist, you're going to slow down all of the inputs, and they're not going to be very helpful.
Go back to the previous slide. So is the effect of the AEA on the presynaptic glutamate neuron antagonist or agonist? Let's see.
The anandamide is an agonist.
Right. Okay. It's having an agonist effect, which, got it. Thank you.
Yeah. Okay. So this goes into a lot of details, as you just heard. And if you want to pursue this more, Nigel, Mark Whiteman, and I wrote a review on all this stuff about 2 years ago now. And you can go into great detail on that. And so some of this feedback that changes the cortical inputs to the stratum is based on cannabinoids, as we were just talking about. Some of it also is based on adenosine. It's not limited only to the indirect pathway. And so you can go into great detail on that. The last of the vignettes on do synaptic alterations really change behavior is the newest one. And we don't really understand it yet, but it's very exciting. And in some ways, it's the most striking of all these behaviors we've seen so far in higher animals. And this is Adrian Stanley, who is a PhD candidate. And he's studying safety learning, which at PI, Peter Balsam looks at this, Eleanor Simpson, probably some other labs are looking at this as well. And in this form of learning, it's classical learning paradigm, the animal's shocked, but it learns when there's a sound that there's no shock. And so the animal very rapidly, and I mean, by rapidly, I mean, Adrian does this 4 times in one day, then the animal sleeps, he comes back the next day, and the animal's already learned the behavior. So it's very fast. And the animal has to sleep on it, which is something that I think is going to be interesting. So here's Adrian's video. Come on, here we go. So the animal was frozen. I was thinking it wasn't moving. Yeah, it wasn't moving because it was freezing. Because now when the sound starts, which is over here, the animal starts to move around. And you get this response in this so-called GO or D1 direct pathway, just as you would predict if you were Dr. DeLong. So there is a massive response to the safety cue. So low activity of this spiny neuron, of these spiny neurons, but then after the cue, and this again happens just after 1 day of training, only 4 times, right after the cue, you get this big response. Remember, the dopamine didn't show us a response with cue and Pavlovian learning, but here the D1 under this one is. And then there's additional activity that's happening until the sound goes away and then starts to dip down again. This is consistent with the idea of a GO pathway. This is entirely novel. And you can look at it with color coding over here. Even at the second day, as soon as the sound comes on, boom, you get this activity. And Adrian noticed this comes in 2 phases, as I mentioned. The one right after the sound begins, and the other as the sound continues and the animal walks around and explores. And you could see how striking this is. This is day 1 when the animal's not learned this. This is day 2. Each of these is a mouse. And you could see the very striking effect of learning in this activity. Now, what's amazing to me, and I think Adrian too, is let's go back to that idea of mouse anesthesia. Light anesthesia. Now, here he's recording from one of those mice that is walking around exploring the cage, who has not learned anything. That's in gray. And you could see there's, you put on a sound, there's a little bit of effect, but very little, it's noticing the auditory inputs. Now, under anesthesia in blue, he plays the sound again, nothing, no effect. Now, after training, and again, just a day training and sleep, this is what happens in a mouse that's walking around. You just saw this. You get this increase, this averages for multiple mice. And, but now you've anesthetized the mouse in the blue. You play the sound. Look, that response is still here. This response that is allied with walking around and exploring goes away in an anesthetized mouse. This one does not. We have a response that's learned so quickly, and it is so powerful that it happens even when the mouse is unconscious. Now, we don't know how this immense change in stimulus response occurs, and that will be Adrian's thesis, we hope. He's determining right now which inputs are changing, and then the mechanism. So I'm still on, let's see how we're doing for time. Okay, I'll try to finish in under 10 minutes if I can. This is what we've shown you so far. The changes in synapses really do underlie changes in behavior in higher animals. First, the story from Avery on Pavlovian and operant conditioning. It shows that dopamine filtering of the cortical inputs can change. In sensory motor behavior, Minerva Wong's story, how the cannabinoids are retrograde are involved in this to filter those inputs. And finally, this new mysterious one, but that there is a extraordinary change with learning in the response to auditory cues. And the mechanism for this is in progress. So the second part with this is going to be much shorter, but it's, I hope, if anything, more interesting to psychiatrists, because we're going to talk about, now that you've heard a lot about these synapses, how these can have implications for psychiatric disorders. And for the time, I'm concentrating on neurodevelopmental disorders. So although people don't always think about the stratum being involved in these, of course they are. Nice review by Mark Fuccello on this. As you all know, ADHD is treated with psychostimulants, which are working strongly in the stratum. Autism, the seizures that we measure in the cortex are also happening in the stratum. Schizophrenia, of course, is being treated with D2 antagonists, OCD, with other compounds that are working on the stratum. Our lab follows a very simple rule here, which is to study developmental disorders, examine development. Why this is not done more universally, I cannot tell you. So we actually have quite a few of the studies on this. I'll mention them rapidly. Gaomei Tang, now an assistant professor in neurology, has a very nice story on how normally in development, both in mice and in people, there is synaptic pruning in the cortex. This is due to changes in the so-called mTOR and autophagy pathways. This is absent so far in all of the autistic kids who have been examined, despite we know that autism is really not a very good term in that it's the whole spectrum of disorders. Her postdoc, who also works with me and with Jim Goldman, Wudu Lado, has a very nice story now, it's not published yet, on the loss of the GABA switch, which is sort of the classic physiological change in maturation of the brain. GABA in many neurons starts off as being excitatory, then becomes inhibitory. And this apparently is happening in our tuberous sclerosis mouse models, and Wudu has a very nice story on this. They also don't mature normally. Now, Avery and Ori, and then Mike Post, who's a postdoc in the lab, have some nice stories just about ready to come out. You can read them on bioRxiv on changes in dopamine release. I'll show one slide on that, and changes on the cholinergic activity that Jeffrey just brought up. So that should be published in a month or two, and it's already on bioRxiv. Now, Ori Lieberman, who just received his PhD in November, and he's back in the clinic now, he's an MD PhD, has some very nice papers on the effects. Remember that spiny projection neurons start off by being overly excitable, and then with maturation, they become very, very unexcitable. And he's determined how this works. The indirect pathway is published this year, or last year now, a few days ago, in eLife, and read that paper. And here's the one that I'll be able to talk about today. It's one from 2 years ago that already published in Neuron, and it's about how dopamine regulates the change in activity of these spiny projection neurons that we're discussing today. Now, all of these list a lot of stuff, right? And it's hard to imagine all this stuff and keep in your head, but here's the way you can do it. Each of these changes, even though they're different mechanisms in different neurons, end up leading to decreased excitation between the time of infancy and childhood. So in other words, you got this critical period where you're learning to look in your mother's eyes and you're learning your language and so on like that. And then things become less excitable. Maybe learning becomes harder, but the trade-off is your personality and what you have learned become more stable. So I'm going to show you essentially one example, and this is from Ori's paper that I just spoke about. This is a weird one because you always think of loss of dopamine as causing Parkinson's and Parkinsonism, and indeed it does. But here, in development, it's quite different. And this is normal animal with this is dopamine inputs in the stratum. This is a spontaneous mutation in the loss of a transcription factor called PIDX3. These animals also have retinal problems. They're called phakia mice. Now, normally, here's a normal mouse, a wild-type mouse, walking around the cage. If you have dopamine loss developmentally, not late in life, you become hyperactive, as you can see with this animal that's running around much faster than the wild type. Does this happen in people? It probably does. There's not that many ways to test in people, but this is very timely. So 1921, people were examining the 1918 flu and the encephalitis that was associated with this. In older people, as you know from our ex-faculty member Oliver Sacks and Awakenings, you know, they would get this encephalitis and later they would develop Parkinsonism. Younger people who had this, and this, you can read this in the 1921 paper, are hyperactive. And then they're very rare diseases, this one, where people don't develop dopamine early in life, and these kids are often hyperactive as well. So very different if you lose dopamine in maturity and if you lose it in early development. So Ori's hypothesis was, well, maybe this transmission is, remember, these animals are running around, activation of the GO pathway. Maybe it's changing these direct pathway inputs. So to look at that, he's examining mice, and mostly he's looking at starting around P10, even before the animals have opened their eyes, up to when they're starting social play and puberty, and then up to adult. So we'll be looking at that system. So the first thing he and Avery realized was there was a tremendous change in the amount of dopamine release. And that is, here it is at day 10, and here's the same stimulus at day 28, and you can look at the change over time. And this is due, and Jeffrey brought this up, this is actually due to changes in nicotinic acetylcholine, and it's due to the changes in the activity of that neuron, which just because of time, we won't go into it, you can read about it. Now I wanna remind you that in adults, these projection neurons are silent unless they go into this up state. And that this is due to this particular potassium channel, which is the major conductance when the animal, rather when the cells are in this downstate. It's called the inward rectifying potassium or KIR channel. So this also, this activity also, as I've mentioned a few times actually, happens developmentally. This is Jim Tepper's work. He's at Rutgers, and Jim was recording from these neurons. And if you look at day 10, you could see they don't have up and down states. They have like an action potential. Let me wait a while, I'll have another action potential. Now let's go up to day 28. Now they are much more hyperpolarized. They're at this down state of -70, where you cannot get an action potential. You go up to an up state, now you get this burst, which again is the language of these spiny projection neurons. So that's what Jim showed developmentally, and Ori is going to show you how this works. So he's going to record by electrophysiology from the cells. So he makes an animal that has red D1 dopamine direct pathway neurons, and he does this in both wild type animals and then ones that have never seen dopamine. And what happens? So you can measure the activity. Let me try to go through this pretty quickly. So normally there is a maturational change. The resting membrane potential, I already mentioned this in passing, becomes deep, typically is hyperpolarized. In the knockout animal, that maturation never happens. Typically, you don't have this current, so you have a this amount of input resistance. You actually get lower input, you get higher input resistance because if you have the KIR channel expressed. Rheobase is just a silly term that us electrophysiologists use. It just means the amount of current you need to inject to get the first action potential. And you can see if you inject the same amount of current, you get many more action potentials in this dopamine knockout animal than in the wild type animal. And you can measure these changes over time the same way that Jim measured them by looking at the number of action potentials. And what you'll see here, and I know I'm doing this quite quickly, but we only have a couple more slides, that if you stimulate a wild type animal as an adult, you'll, in these conditions, one action potential, here a whole burst. And you can look over the change in age, and you see that the resting membrane potential never hyperpolarizes in animals that haven't seen dopamine. They don't develop this change in input resistance, and in sum total, when you measure from these neurons, in a normal wild type, as I've been telling you a lot today, there's very little activity, but in the knockout, they're depolarized and boom, you get some activity. You get it again and again and again. No wonder these animals are running around the cage. So what does this do to? Ori did some very nice work where he realized that he could measure the amounts of cure current, and they do this because cesium will block this particular current. You can measure it, and you see this increase in this potassium inward rectifying current with age. By around day 28, the normals have maturity, but the ones that have never seen dopamine never get this maturity. They never get this maturation. This is because they don't have this current. They are never able to mature, and they stay overactive throughout life. Now we can replace the dopamine by giving L-dopa in the chow. This is with Eugene Moshoff in our group who figured out how to do that. And you can give them chow at different periods in development. And by doing this, you could see where you can make them, where the dopamine will help with the maturity. And just, I think this is the next to last data slide. And just to say this rapidly, we find that you can, so here the normal currents in a wild type, they're pretty much absent in a knockout. If you give them L-dopa, you can get these currents, these cure currents back, and now you get a normal maturation of that mouse. This only happens if you give it to them when they're juveniles. If you give it to them when they're adults, it's too late. And we don't know if this has anything to do with treatment of, say, Adderall and Ritalin in kids, but it is something that if we could figure out how to design the experiments appropriately, is obviously something that should be looked at. Okay, last data slide, how does this work? For time, I won't go into it very much, but the cure currents require a cofactor, which is a lipid called PIP2. And, or you can record from the cells, put in some water-soluble PIP2 that has no effect on the cure currents in wild type, but it starts to restore the normal current to the knockout animals that have never seen dopamine. And if you do that with the L-dopa and the chow trick that we told you before, you get a complete recovery to this kind of normal activity again. So what dopamine is doing is it's activating through a series of second messenger systems, and it's a slow effect, the activity of PIP2, but can only do it during this little critical period of synaptic plasticity. Okay, so I'm going to summarize. This is the last stuff. Dopamine is required for decreased excitability of one of these output pathways, the direct pathway, the GO pathway. Without it, the direct GO pathway is hyperactive, consistent with what you saw in that mouse behavior and those rare cases that we understand of people that develop with very low levels of dopamine. The dopamine action on the direct pathway has temporal specificity. You can normalize it, but it has to be in this little pocket of early development. Adulthood, unfortunately, is too late. The critical period closes when dopamine no longer can couple to the increasing the appropriate levels of this lipid in the dendrites. So this is what I've spoken about today. I know it's a lot of stuff, but I hope it's interesting for the audience. I hope to have shown you that synaptic changes genuinely do alter behavior in higher animals from the 3 vignettes from the graduate students, and shown you a little bit about our work in neural development, but one that is obviously quite directly related to these synapses and how this could have implications for neurodevelopmental disorders, in this case, particularly the ones that people in this audience are treating with dopaminergic agents. Finally, and this is just to remind you that there's actually quite a bit going on with the maturation here. Acknowledge the people in the lab. I believe everybody who's in the lab is doing fantastic work. I only spoke about some of it. We're doing a lot of other work in mTOR pathways and neuroimmunology and new ways to measure synapses and so on. And I mentioned Avery, who's here at Santa Claus hat, Maha, who you'll see walking the halls, so say hello to her, Ori, who's now back in the clinic, Adrian, who's in the lab over here, Stefano, and Mike. Here's Stefano, and where's Mike? And there's Mike. I spoke about some former members of the laboratory, all who have gone on to great things. And these are, we work with quite a few collaborators, and these are the ones I mentioned specifically. So thanks for inviting me again on this beginning of the year, an auspicious historical day. I'm as curious as anybody is here to look what's happening in the news right now. And sorry to go over a bit, but it's simply due to my own excitement at the great work that these people are doing. Okay, happy to take any comments and questions.
Thank you, Dave.
There's a lot of chats in here, which I couldn't see when I was on before. Shall we start with that? Paul Warren said, we can see him and hear him. Thank you.
Yeah, most of the chats were while we were setting up, but we have a question from Nen Nunez. Has anybody tried giving L-DOPA to hyperactive children?
I'm going to say I have never, and I've done a lot of work with L-DOPA. I've never seen anybody do that. Does anybody else know?
Yeah, I can address that question. So L-DOPA has been tried to some extent in ADHD with sort of middling results, and certainly it's much less effective than psychostimulants.
What age, John? Because I think that that's going to be, Jonathan, sorry. According to our work, it's got to be early.
Yeah, it's a good question. These were trials done decades ago. I'm not sure. I'd have to go back and look.
Yeah, it's an interesting point, Ed, and it might be something we'd want to consider. You know, conceptually, why should this be different than a dopamine releasing agent like Adderall or a dopamine reuptake blocker like normophenzine or Ritalin or something? And I would have to say that that's not exactly obvious to me right now. I know why it would be different in a Parkinsonian patient because their dopamine axons are gone and we're building up dopamine in their surviving serotonin neurons, which are still alive. But why these different things might work different on youth hyperactivity, I can't really say. I think that maybe it's worth some consideration.
Dave, I wanted to make a comment. You very correctly pointed out that finding children who have grown up in a hypodopaminergic state is not easy. But one possibility, I don't know if you've considered this, is kids with phenylalaninuria who they're caught very early on in life, and they're immediately put on a diet that restricts any phenylalanine or tyrosine intake. And so by default, they're going to grow up hypodopaminergic. And I believe it is associated with hyperactivity.
Uh-huh. Interesting.
And these are kids that are screened.
Yeah. Huh. Okay. This is almost certainly working through the D1 system. And what's the status on D1, approved D1 agonists in the clinic? None, right? Or is there anything? That might be something to consider also. Because this is happening through D1. It's not happening through D2. I don't think you just want to give them, you're not going to want to give them antipsychotics. You're not going to want to give them L-DOPA. I think you're going to want to give them something pretty specific. Jeffrey, you're saying something?
Yeah, the pharmacodynamics are a little bit difficult to reconcile. If you have indirect dopamine agonists like stimulants being effective, and then you have a precursor not being effective, that's one thing. The other is that for D1, there haven't been really many viable D1 selective agonists for whatever the medicinal chemistry.
I mean, we got them some in the lab, but I don't think they're proof.
No, there's one right now that's under investigation, and we're doing the study in schizophrenia. And so
an agonist, though, not an antagonist.
No, and it's an agonist that has high intrinsic activity and also is tolerable at doses achieving this adequate level of occupancy. It's a Pfizer drug that they gave to this new company, Cerevel. And so it's something that, Jonathan, you should jump on. I can facilitate the contact with Jonathan Javich.
Well, Jeff, sorry to interrupt on this, but I want to make this point. So this is going to be, if you did the D1 early, this would be very, very different than the psychostimulants that presumably people have tried in these kids. So if there was a safe way to try this out, and it would have to be done early. I'm afraid if we're going to take our basic work and say that has any relevance to this, it's not likely to work, say, after the age of 6 or 7.
We can talk about it. It might be interesting to try
what's the
the
heritability
compounds in
of
Dave's
ADHD?
early animal model to try to see if it would work just like L-DOPA. If you
That's a good point. We could try it. We could try it. We could try it
first in your developmental model and then think about it.
Yeah, I think that's a very good idea, Jonathan. And it's not so hard. You actually don't even need the electrophysiology, I think. You just have to look at the behavior and see if you can inhibit that hyperactivity in the mouse by using a D1 agonist. So I think that's, other Jonathan, I think that's a very good, that's a very creative suggestion, and it's worth a try. It's not really that hard to do in mice.
Jonathan Javich, did you ever discuss this with David Gray?
No, because we were always focusing on adults and schizophrenia, but it'd be interesting to try to bring this in at an early time point. So we'll talk about that.
Jeff wants to know how it works on Guillermo's model psychosis. Okay, Guillermo.
But before we do that, you got a series of other questions. So
I only see the ones on the chat.
There's actually a question and answer as well as the chat. So the question and answer is the relevant one right now.
Okay, gotcha.
The Mosen has a long question here, which I will first. He complimented you. Terrific talk going from channel biophysics, synapses and physiology, to behavior and neuropsychiatric disease. So you got a good compliment. He wanted to go back to the synuclein knockout.
But for Mosen, it counts. The compliment. But from him, I'll take the compliment.
You can read it later. All the compliments. He's asking a question about the synuclein knockout experiment, saying that is it really that it's a deficit in facilitation, or is it that because it looked to him like there was more dopamine released on the first pulse? So could it be that you actually have a higher release probability and then a ceiling effect rather than not going up?
So Mosen has very good eyes. So that Mosen, you're correct when it comes to the cholinergic system. That you get more of an activation because the first pulse is less. That's not true with the synuclein. The first pulse is the same. So it is a genuine facilitation, and it is not classic facilitation. I didn't go into this due to time, but Mike Post in the lab measured GCAM from these guys. So classic facilitation would have to do with so-called residual calcium, a little bit of calcium that's left over from the previous pulse. That is not the case here. The pulses are too far away. In this case, they're about 6 seconds. They've gone back to normal calcium. There's no residual calcium. This is actually due to an effect of alpha-synuclein on fusion. It's a different thing.
Mosen also wanted to know what's missing in the math model that doesn't account as well for the decay kinetics, but I'm not sure that's something we should
really, this kid. I'm sorry, Mosen, I'm calling you a kid. You're not a kid. I was on your PhD committee, but you're still on. That you do have good eyes. So that decline is not biological. When you run cyclic voltammetry and not constant potential amperometry, you get a buildup of fried dopamine on the electrode site. And so that delays the dopamine from coming on and off the electrode. And so if you run a bunch of stimuli, you will start to see that in cyclic voltammetry. You do not see that in amperometry where the dopamine is fried and becomes a quinone and leaves to never build up. So actually one of Maha's important experiments is to do the same thing with amperometry. But what you noticed is not biological. It's sort of a quote artifact. It's not really an artifact, but it's a property of an electrochemical electrode. Good eyes.
Jeremy also complimented you, so that counts too. Lovely.
That does. Okay, thanks, Jeremy.
So Jeremy's asking about the potential opposite effect. What would be the effect of too much dopamine early in development rather than too little dopamine in early development? Whether you've looked at that with the same approach.
No, we have not looked at that. And I do know that if you give more L-DOPA to a young animal, it does not increase the maturation, or in other words, the ability of the brain to become more silent, specifically the direct pathway neurons, but the whole circuit. So I would say on the basis of Ori having administered L-DOPA to young animals, and also Avery's done a bit of that too, I would say so far we have not seen anything, but as with all experimental science, maybe we're not asking the right questions. We haven't seen any maturational change that we have noticed.
It's making me think about Christoph Kellendong's work, where it's not increasing dopamine, but it's increasing presumably dopamine signaling by overexpressing D2s early in development. And that actually does have substantial effects.
If we give too much L-DOPA, it's essentially for certain. I could even show you some data from that I showed you very briefly from Minerva. What you get is somewhat of a desensitization of at least of the D2 receptors. So that should have functional consequences. So Jeremy, good question. All I can tell you is too much L-DOPA in normal animals does not seem to do anything. And my understanding is that is also pretty much valid in people.
Dave, I had a question about the PIP2 at the end. Not sure I took it in properly, but if you give this effect only works for the L-DOPA you said during development, and that you said that works through PIP2, and that PIP2, you said, I think that PIP2
it's not a rapid action.
Is inactivated. So will PIP2 fix it in adulthood? Is the problem that L-DOPA doesn't lead to enhanced PIP2 in adulthood, but if you exogenously put it on, it will fix it?
Yeah, so Ori, if you're still on, he's really the right person to answer this, so invite him up if he's there. But if you take one of those hyperactive animals that didn't grow up with dopamine, and you record from the neuron, and now you put in water-soluble PIP2, that will fix it. So yes, you're correct that you should be able to, if you can figure out how to do that, you would have a treatment.
So the channels are expressed normally, but they remember whether they got PIP2?
No, it's not memory. The PIP2 has to actually be there. It's actually genuine cofactor.
Why doesn't PIP2 get activated during adulthood? So why if you fix that during development and get PIP2, why do the channels then end up being different in adulthood? I don't get that.
I don't think the channels are different. I think it's literally the availability of PIP2 to the channel. And you can replace that because, in fact, I showed it to you. Put water-soluble PIP2 into the recording electrode, you start to get the cure becomes activated. This, by the way, is very different in the indirect pathway, where the cure channels are actually that's an mTOR autophagy related mechanism, and you simply don't have them where they can be activated appropriately. But in the D1 pathway, it appears to be PIP2. If you are able to get PIP2 activation of the channel, you would normalize the direct pathway.
Well, this last one I gotta read to you, man.
I'm sorry?
Do you remember Patricia Liebens?
Oh, Pat is from my hometown.
Yes, she knew.
Carbondale, Illinois.
Am I supposed to read
Pat,
this?
and actually, yeah, nerdy curly-haired runt. Well, that's actually true. I don't know. And Pat's younger brother, Bill, was my best friend growing up in the small town in Illinois.
That's funny.
And she says, I'm a Yale-affiliated child and adolescent psychiatrist in New York. All right, Pat, that's great. Thanks, Pat. Hometown, go Salukis.
So any other questions? I don't know if Guillermo's here and wants to try to comment, but I don't know if he can comment without Simon's permission other than by typing.
Yeah, I think Simon would need to bring Guillermo into the panel, if that's possible.
Guillermo, if you're here and want to be brought in, chat or question.
Yeah, also Ori, if you're still on and you want to address Jonathan's question in more detail, go ahead.
I think we only have 2 or 3 more minutes, but
okay,
we could do one more thing. But I think you really did demonstrate the incredible breadth of the work and all the contributions. So
thank you. Yeah, I don't know if Martha Crowner is still around. She went to grade school with me too, from Carbondale. So we had a few people from this little town that ended up at Columbia for some reason. Okay, I have the feeling, am I still sharing?
No.
Okay.
No.
All right, I think I probably confused everybody. I'll just
Maybe
mention
everybody wanted to
people
go
that are
watch, look at the
interested in
news.
this kind of
like you
work.
mentioned.
Sorry, Jonathan, if people are interested in this kind of work, they can email me and we'll put them on the Friday pandemic lab meetings where we go into the stuff in great detail. Sorry, go ahead.
No, I was just suggesting that maybe everybody really does want to go read the news like you were saying.
Okay. Yeah, what happened? I haven't been able to
I'm
look at the
listening
news
to you.
while I'm speaking.
So thanks very much, Dave. Everybody have a great day.
Okay, thanks. Bye now.
Much,
Bye-bye.
Dave.